pharmaceutical impurity testing plays a critical role in ensuring the safety, quality, and effectiveness of medicines. In this presentation, we will explore what pharmaceutical impurities are, why impurity testing is so important, and how it is carried out in real pharmaceutical laboratories. Every medicine contains more than just the active pharmaceutical ingredient, or API. Along with the intended drug substance, there may be small amounts of impurities that are invisible but powerful. These impurities are not always obvious, but if they exceed acceptable limits, they can significantly impact patient safety and product quality. This is why impurity testing is a critical part of pharmaceutical development and manufacturing. An impurity is any unwanted chemical present in a drug substance or drug product that is not the intended API or excipient. Impurities can originate from different sources. They may form during chemical synthesis, appear during storage, or develop as the drug degrades over time. Even very small quantities of certain impurities can pose serious risks, which is why strict control and monitoring are essential. Pharmaceutical impurities are generally classified into several categories. Organic impurities include starting materials, intermediates, byproducts, and side reaction compounds. Inorganic impurities may come from catalysts, heavy metals, or inorganic salts used in manufacturing. Residual solvents are organic solvents that remain after processing. Degradation products form when a drug breaks down due to factors such as heat, light, moisture, or oxygen. Each type requires a specific analytical approach for proper evaluation. Impurity testing is essential for multiple reasons. First and foremost, it protects patient safety by preventing exposure to harmful substances. It also ensures regulatory compliance with global health authorities. In addition, impurity testing supports product quality and stability throughout the shelf life of the medicine. Failure to control impurities can lead to batch rejections, recalls, and long-term damage to a company's reputation and brand trust. Regulatory expectations for impurity control are largely defined by the International Council for Harmonization, or ICH. ICA focuses on impurities in drug substances. ICH-Q3B addresses impurities in drug products. ICH-Q3C provides guidance on residual solvents. ICH-Mizidin covers mutagenic impurities. Regulators establish identification and qualification thresholds, requiring manufacturers to detect impurities, identify their structure, and demonstrate that they are safe at specified levels. Detecting impurities requires advanced analytical techniques. High-performance liquid chromatography, or HPLC, and ultra-performance liquid chromatography, UB, are widely used for organic impurities and degradation products. Gas chromatography is commonly applied for residual solvent analysis. LCMS and GCMS are powerful tools for identifying unknown impurities. ICP-MS is used to detect elemental and metal impurities. In practice, impurity testing often involves a combination of these techniques. Impurity profiling goes beyond routine testing. It is a systematic process that involves detecting impurities, identifying them, quantifying their levels, and controlling them throughout the product lifecycle. Forced degradation studies help predict how impurities may form under stress conditions. Stability studies monitor impurity growth over time and support shelf life and storage recommendations. In real pharmaceutical laboratories, impurity testing presents several challenges. Unknown impurities may appear without reference standards. Some impurities exist at extremely low levels, requiring highly sensitive analytical methods. Method development and validation can be complex, especially when regulatory expectations are strict. These challenges require scientific judgment, risk-based decision-making, and clear communication with regulatory authorities. Effective impurity control starts with preventing impurities from forming in the first place. Pharmaceutical companies optimize manufacturing processes, carefully control raw materials and suppliers, and validate cleaning procedures to avoid cross-contamination. Ongoing monitoring through stability testing and trend analysis helps ensure consistent impurity control. Strong impurity management reflects a robust pharmaceutical quality system. Impurities are an inevitable part of pharmaceutical manufacturing, but they are manageable. Through proper testing, monitoring, and control strategies, impurity testing protects both patients and products. Successful impurity management requires a strong combination of scientific understanding and regulatory compliance. When done correctly, pharmaceutical impurity testing ensures that every dose delivered to patients is safe, effective, and of high quality.